Hardest Praxis 5435 Practice Questions. Step-by-step solutions. Preparation quiz. Question 1. A student is studying the process of meiosis and understands that it produces gametes with half the number of chromosomes as the parent cell. The student asks the fundamental question: why is this reduction in chromosome number necessary for sexual reproduction? Which of the following explanations best answers this question? Is it? Option A. Meiosis ensures that offspring have the same number of chromosomes as their parents by having the chromosome number in gametes, so that when two gametes fuse during fertilization, the original diploid number characteristic of the species is restored. B. Meiosis is primarily important because it increases genetic diversity through the mechanisms of crossing over and independent assortment, which is why offspring resemble, but are not identical to their parents. C. Meiosis allows for the selective elimination of harmful mutations by creating gametes with different combinations of alleles, ensuring that only the healthiest combinations are passed on. Or D. Meiosis is necessary to produce haploid cells that can then undergo a series of rapid mitotic divisions to form a new multicellular organism. The correct answer is A. This question addresses the core purpose of meiosis in the context of sexual reproduction. The primary and most essential function of meiosis is to reduce the chromosome number by half, producing haploid gametes, sperm and egg. If gametes were diploid, the fusion of two diploid gametes during fertilization would result in a tetraploid zygote, and the chromosome number would double with every generation. By having the number, meiosis ensures that the fusion of two gametes restores the diploid number that is characteristic of the parent species. Option A correctly states this. Option B describes an important consequence of meiosis: genetic variation, but it is not the primary reason for the reduction division. Option C is not a primary function. Option D is not accurate. The restoration of the diploid number is the key concept. Remember, we can take your Praxis 5435 exam for you remotely and undetected with 150 score guaranteed. Message us via the number in the bio. Question 2. A student is studying the periodic table and notices that elements in the same group, the vertical column, tend to have very similar chemical properties. The student asks why this pattern occurs. Which of the following explanations best answers this question? Is it? Option A. Elements in the same group have the same number of occupied electron shells, and this determines how they interact with other atoms. B. Elements in the same group have the same number of valence electrons in their outermost shell, and these are the electrons involved in chemical bonding, giving them similar reactivity. C. Elements in the same group have the same atomic mass, and atomic mass is the primary factor that determines chemical behavior. Or D. Elements in the same group were all discovered around the same time in history, and were grouped together based on their discovery dates. The correct answer is B. This question addresses the fundamental organizational principle of the periodic table. The periodic law states that the chemical and physical properties of elements are periodic functions of their atomic number. This periodicity is explained by the electron configuration of the atoms. Elements in the same group have the same number of electrons in their outermost shell, known as valence electrons. Valence electrons are responsible for forming chemical bonds, so elements with the same valence electron configuration exhibit similar chemical behavior. Option A is incorrect because the number of electron shells increases moving down a group, not within a group. Option B correctly identifies valence electrons. Option C is incorrect. Atomic mass increases down a group, but is not the primary determinant of chemical properties. Option D is historically inaccurate. The periodic table's power lies in its ability to predict chemical behavior based on electron configuration. Question 3. A student is studying the process of photosynthesis and learns that plants release oxygen gas as a byproduct. The student asks a historically important scientific question. Where does this oxygen come from? Does it come from the carbon dioxide CO2 molecule or from the water H2O molecule? Which of the following types of experiments would best demonstrate the source of the oxygen produced during photosynthesis? Is it? Option A. Grow plants in an atmosphere containing carbon dioxide where the oxygen atoms are labeled with a heavy isotope, and in a separate experiment, grow plants in water where the oxygen atoms are labeled. Then, analyze the oxygen gas produced to see which set of experiments yields labeled oxygen. B. Measure the rate of oxygen production under different light intensities to see if it correlates more strongly with the uptake of carbon dioxide or with the availability of water. C. Compare the total mass of oxygen produced by plants grown in water versus plants grown in soil to see which environment yields a higher rate of oxygen evolution. Or D. Measure the increase in the dry mass of a plant over time and correlate it with the amount of carbon dioxide absorbed from the atmosphere. The correct answer is A. This question references the classic experiments by Samuel Rubin and Martin Kamen in the 1940s that used isotopic tracers to solve this puzzle. They used a heavy isotope of oxygen, oxygen-18, as a tracer. They grew photosynthetic organisms in an environment where the water contained oxygen-18, and separately in an environment where the carbon dioxide contained oxygen 18. They then analyzed the oxygen gas produced. They found that the oxygen gas was labeled only when the water was labeled, proving definitively that the oxygen released during photosynthesis comes from the water molecules, not from the carbon dioxide. Option A correctly describes this type of isotopic labeling experiment. Option B measures rate, not source. Option C is not controlled. Option D measures carbon fixation, not the source of oxygen. This experiment is a classic example of using tracers to follow atoms through biochemical pathways. Question 4. A student is studying the human nervous system and learns about the structure of a neuron in the synapse, the gap between two neurons. The student asks how an electrical signal in one neuron manages to cross this physical gap to stimulate the next neuron. Which of the following explanations best describes the most common mechanism of synaptic transmission? Is it? Option A. The electrical signal or action potential simply jumps across the synapse as a spark, directly stimulating the next neuron, a process made possible by the close proximity of the cells. B. When the action potential reaches the end of the first neuron, it triggers the release of chemical messengers called neurotransmitters. These molecules diffuse across the synaptic cleft and bind to specific receptors on the second neuron, causing ion channels to open and generating a new electrical signal in that neuron. C. The axon of the first neuron physically fuses with the dendrite of the second neuron, creating a continuous pathway so the electrical signal can travel without interruption. Or D. Calcium ions carry the electrical charge directly from one neuron to the next across the synapse, acting as the primary signaling molecules. The correct answer is B. This question tests knowledge of synaptic transmission, the fundamental process by which neurons communicate. At the vast majority of synapses in the nervous system, communication is chemical, not electrical. When an action potential reaches the presynaptic terminal, it causes voltage-gated calcium channels to open. The influx of calcium ions triggers synaptic vesicles to fuse with the presynaptic membrane, releasing neurotransmitters into the synaptic cleft. These neurotransmitters diffuse across the gap and bind to specific receptor proteins on the postsynaptic membrane. This binding causes ion channels to open, leading to a change in the membrane potential of the postsynaptic neuron, which can generate a new action potential. Option A describes electrical synapses, which exist but are less common. Option B correctly describes chemical synaptic transmission. Option C is incorrect. There is a physical gap. Option D misrepresents the role of calcium, which is an intracellular trigger, not the carrier of the signal across the synapse. Understanding this process is key to neurobiology and pharmacology. Question 5. A student is studying the properties of waves and learns about the relationship between frequency, wavelength, and speed. The student knows that the speed of light in a vacuum is a constant. If a beam of light has a frequency of 6 times 10 to the 14th power hertz, what is its approximate wavelength, using 3 times 10 to the 8th power meters per second as the speed of light? Is it? Option A. 5 times 10 to the negative 7th power meters, which is 500 nanometers. B. 2 times 10 to the 6th power meters. C. 1.8 times 10 to the 23rd power meters. Or D. 5 times 10 to the 7th power meters. The correct answer is A. This question requires the application of the basic wave equation, which states that speed is equal to frequency multiplied by wavelength. To find the wavelength, the equation is rearranged to wavelength equals speed divided by frequency. Given the speed of light c is 3 times 10 to the 8th meters per second, and the frequency f is 6 times 10 to the 14th hertz, the calculation is 3 times 10 to the 8th divided by 6 times 10 to the 14th. This simplifies to 0.5 times 10 to the negative 6th, or 5 times 10 to the negative 7th meters. This value, 500 nanometers, falls within the visible light spectrum. Option A correctly shows this calculation and result. Option B and D yield wavelengths that are many orders of magnitude too large. Option C is also an impossibly large number. Understanding this relationship is fundamental to working with the electromagnetic spectrum. How many did you score out of 5? Let us know in the comments below. Also, tell us which exam we should cover next. If you found this video helpful, please like, share, and subscribe for more.